Coming to the distal control elements, the distal control elements are generally, there are two types of distal control elements. They are called as enhancers and silencers. These enhancers and silencers, they are seen, as the name suggests, as I said in the previous video, they are seen, these sequences are seen far away from the gene to be transcribed, but they will be they are influencing the activity of the gene. Such elements are called as the distal control elements. One distal control element is enhancer, the other one is called as silencer. So what enhancers and silencers do is, enhancers are bound by proteins called as activators, silencers are bound by proteins which are called as repressors. So enhancers are DNA sequences, silencers are DNA sequences, activators and repressors, they are proteins. Activator, this is a protein, and this repressor, this is also a protein. These are DNA elements. This is also a DNA element, which is silencer. So what the silencers and repressors and silencers do is, what they actually do to control the gene. So I take a particular gene. We have the gene starting from here. You have the promoter site here, and I'm taking a distal one. So take it that these particular elements are far away from this. So this is a particular gene region. You have the promoter here, and now I am having an enhancer element. So this is the enhancer element. So now what happens is, as we said earlier, the enhancer element will be bound by a protein. So if it is bound by a protein, this protein will be called as an activator protein. So whenever the enhancer element is bound by the activator protein, what this enhancer does is, when the protein binds, this protein will cause the particular DNA to loop in the sense that after this activator binds, this will be like this. This will be the promoter region. This is the gene which is shown above. Now this enhancer region and activator region will be like this. Now this activator protein which is bound to this enhancer element, so this is the same thing. So when the activator protein was bound, the enhancer is now looped out. It was far away from the promoter region. Now it has looped and it has reached the promoter element to regulate the promoter. So when the enhancer element, when the activator protein is bound to the enhancer, it will loop and reach the promoter, and it will activate transcription. So enhancers activate transcription, and when the other so if you're having, instead of this one, if you're having if we take the other one, which is our silencer. So if this is a silencer element and a repressor protein binds here, so the same thing happens. So here is a protein which is called as repressor. Repressor protein, the same looping occurs. Silencer, the protein is a repressor. Silencer is a DNA element. So when a repressor protein with the silencer loops, so when a repressor protein binds, a silencer loops and reaches the promoter region, it represses the transcription of that particular gene. So this is how the distal control elements acts. This enhancer, the enhancer element, the silencer, the activator, repressor, such things are usually asked as 1-mark questions. So now that was about the transcriptional control. We talked about proximal as well as distal. Next is post-transcriptional control. So how can you control this particular pathway or particular the expression of a gene after it is being transcribed? That is what is called as post-transcriptional control. It occurs by slicing. It's not slicing, it's splicing. The spelling, check the spelling there. Splicing and alternative splicing. So splicing is nothing but in a eukaryotic mRNA, the mRNA is not actually translated as a mature mRNA. It is transcribed as transcribed in the form which is to be modified, and then an mRNA is to be formed. A mature mRNA is to be formed. So the mRNA which is formed directly, which is being transcribed, contain two sets of sequences. They are called as exons and introns. So introns are intervening sequences. These introns should be removed. When introns are removed and exons are joined, you get a complete mRNA. So this removal of introns from exons is called as splicing. It is by the process which is called as splicing, the introns are removed and the exons are joined. Alternative splicing is nothing but, if you take, we'll talk about splicing as well as alternate splicing here. I'm taking an mRNA. It will contain many elements. It will have intron. I take it as intron 1. You have exon 1 here. Then you have intron 2 and exon 2. So when a mature mRNA, or after splicing, what happens is this introns will be removed. You will have exon 1 and exon 2. So now the complete mRNA is being formed. So this is what is called as splicing. This is one control whereby an mRNA can be correctively formed. So there is something which is called as an alternative splicing, where you can just grapple this particular protein. That means this is a correct mRNA. That means you get the correct protein. So if this protein is not to be formed inside the cell, it is to be regulated at this particular level. What can be done is that what is called as an alternative splicing, where you can just jumble up the exons. That means in this case you can just jumble up like this, E1, E2. E1, E2 in this particular order from left to right is the correct protein. If I jumble E1, E2, you get a different protein or a useless protein. So this is what is called as an alternative splicing. This is also a mechanism for regulating gene expression. Next is translational control. That means if at all the mRNA is being formed, how can you control at the level of translation? So there are regulatory proteins which are binding to the 5' and 3' untranslated region of the mRNA. So when an mRNA after exon shuffle, the splicing, etc., a mature mRNA is being formed. In that mature mRNA itself, if you take the mRNA from the 5' to the 3' region, the AUG or the correct protein sequence will be starting from here. You will sometimes have an untranslated region in the 5'. UTR means untranslated region in the 5' region, and sometimes it will be present in the 3' region. So anywhere you can see this untranslated region, because they will not be translated. So these are regulatory sites. So regulatory proteins come and bind here. So when regulatory proteins come and bind here, as we talked in translation, our ribosome cannot come and bind to this particular mRNA. So that is how regulatory proteins and this 5' UTRs, they are restricting, or they are controlling gene regulation at the level of transcription. So this is nothing but inhibition. Regulatory proteins bind, they inhibit ribosome binding. The next thing is microRNAs or miRNAs. So miRNAs are produced by the enzymes which are called as disers. miRNAs are usually very big mRNAs which are cleaved by a particular protein or an enzyme which is called as dicer. It cleaves it and it produces a complex. Because it is single-stranded, this miRNAs are single-stranded. As normal RNAs, they will be easily degraded in the cell. So they are protected by a group of enzymes and form a complex which is called as miRNA complex or protein complex. So this miRNAs which are produced will be complementary for the normal mRNAs which is inside the cell. So what happens is, if a particular mRNA should not be translated, the cell produces more and more amount of miRNA which is complementary to that mRNA. They will go and bind to that mRNA. They will form double-stranded structures. That mRNA will not be translated. And mRNA mRNA, which is single-stranded, that is usually translated by the ribosome. When it becomes double-stranded by the binding of miRNA, it will not be translated. So that is the next level of control by miRNAs. The last one is a post-translational control. That means a protein is produced, and now that protein has to be degraded, or the function of the protein is over. An enzyme was produced. For example, the enzyme which we said in the starting of the session, take it as lipase. Digestion of lipid is over. Now lipase is still present in the digestive system, which is of no use. That is to be degraded. That is usually degraded in the cells with the help of the structures which are present in the cells, which are called as proteasomes. Proteasomes are protein degrading organelles which are present inside the protein degrading machines which are present inside the cells. The proteins usually are degraded. A protein which is to be degraded should be differentiated from a protein which is currently used by the cell. So there should be a tag. That means that protein is to be degraded. The tag is another protein which is called as ubiquitin protein. So a chain of ubiquitin, which is called as polyubiquitin, will be added to a protein. So that particular process is nothing but, for say, this is a protein that is to be degraded. So what occurs is, now I am going to add many beads. Consider it as a string like this. So consider a protein. So I am taking a protein, and this protein is to be degraded. Now this will be added by a chain of protein which is called as polyubiquitin. Many ubiquitin proteins will be added. These are ubiquitin protein. Many ubiquitins are added. These are single ubiquitin proteins, and that is what is called as polyubiquitination. And now you have the particular protein degrading structure which is in the form of a barrel with a lid, which is called as the proteasome. Now what happens is, normal protein will not have this ubiquitin tag with it. So whenever a protein reaches this proteasome, if it does not have a ubiquitin, it will not fall into the proteasome. But whenever a protein reaches the proteasome with the ubiquitin tail, the lid will recognize it. So the lid will recognize the ubiquitin and make this particular protein fall into this barrel. This barrel contains many peptidases which will be cleaving off the peptide bonds. So the protein will be cleaved into many amino acids. These amino acids will be recycled. The component amino acid will be recycled. It will be used for the production of, new proteins. So this is the next level of control, what is called as the translation. So in eukaryotic gene regulation, the particular process of from gene synthesis from mRNA synthesis to protein production, it is regulated at different steps. It is regulated at 5 different steps. Chromatin modification, which is the modification of histone. Transcriptional, that means you have promoter elements which are proximal and distal control elements which either activate or repress it. Post-transcriptional, which is splicing machinery. Translational control, mainly by miRNAs, and post-translational with the help of ubiquitin and proteasome. So this is an overall explanation about eukaryotic gene expression. So we have completed gene regulation of prokaryotes and eukaryotes. So coming to another one, the differences. So these differences, you just go through the differences. These we have discussed at different points in this particular discussion. Such a question might be asked. That's why I have prepared such a slide for this. We have 8 different points for this is well enough for a 5-mark question. So this is the difference between the prokaryotic as well as the eukaryotic gene regulations. So thank you. With this, we complete our discussion about in molecular biology topics. We have covered the major topics in molecular biology. So if you have any doubts regarding the any of the topics, we can either sort out in the discussion format in Zoom, or you can either WhatsApp me, or you can put it directly into the Google Classroom. Thank you.